So next we have, and there you are. Hello, Oliver. Dr. Oliver Deusch. Am I pronouncing your last name correctly?
Almost, but very close. That's good enough. Yeah, thanks.
Well, go ahead, please. What's your
Deutsch.
last? Deusch. Okay.
Deutsch. Yeah, but like very, very good. Yeah.
My American dry anyway. So, so very excited to have you. So, Dr. Deusch is the scientific product manager for Computomics. Studied bioinformatics at the University of Dusseldorf. Graduated on evolutionary genomics of plants and algae. You know, so his talk today is on interactive and reproducible metagenomics. So I'll go ahead and turn it over to you. And
yes, thanks. I'll start sharing my screen. Excellent. Can you see my screen?
Yes. We're all good.
Cool. Okay, I just need to, okay, cool. So yeah, thanks for this. So I'm going to talk, I'm not able to, okay, so sorry for this. So I'm going to talk about interactive and reproducible metagenomics data analysis with MEGAN6 Ultimate Edition and MORPHEUS. I'm Oliver Deusch. I'm a scientific product manager at Computomics, and well, okay, it's been a while since my background in genomics in plants and algae, because for the last 10 years almost, I've worked in bioinformatics for the microbiome. So I've developed microbiome solutions and brought them to academic as well as commercial institutions. Okay, so let me start and give you a very brief overview on Computomics, our company. So something isn't working well. Okay, but so this is the company overview. So we were founded in 2012 by field leading experts from the areas of machine learning, plant research, and bioinformatics from institutions as the ETH Zurich, the Max Planck Society, and the University of Tübingen. Our team brings together a combined expertise of over 100 years in bioinformatics and machine learning, and we apply those methods from those areas and our expertise to data sets from all sorts of areas like genetics, phenotyping, the microbiome, and also environmental data sets. Since our founding, we've completed over 130 projects with customers from 12 different countries. So even though we're a company located in southern Germany, our customer base is pretty much international. And we've worked with 8 out of 10 of the largest breeding companies. Our goal is to disrupt plant breeding using our AI methods to create new varieties that can withstand challenges such as climate change. So as you see, a lot of our business is around plant breeding, but today I'm going to talk about the microbiome work we do. And this is a product overview of all our products. And today I'm going to focus on MEGAN6 and MORPHEUS, which are the metagenomics products. But just so you know, I'm going to give a very quick overview of the other products. So we have tools for causal gene identification and gene editing, a platform for working with methylation data and differential gene expression. We have products for genome annotation and assembly, but we also have the main tools are, or the main platforms are for automated phenotyping in a reproducible way. So if you want to phenotype hundreds of thousands of plants in a reproducible way, we've got a solution for that. And we also have a technology that allows, that we use to guide breeders on which varieties to cross to generate outperforming offspring, basically. Okay, but back to the metagenomics that I'm going to talk about today, MEGAN6 and MORPHEUS. So MEGAN6 is the world's interactive metagenomics analysis tool. And so it's now in the 6th version, as the name suggests. And since its release, it's been cited in more than 4,000 scientific publications. So it's a very well-used software. It allows you classification down to strain level. And more recently, long read support has also been added. So if you're working with long reads, PacBio or Oxford Nanopore data, MEGAN can handle this as well. MEGAN comes with a set of command line tools, for example, the Meganizer, and I'll explain a bit what that means in a minute. But the core use is really to visualize and analyze metagenomics data in an interactive way. And for that purpose, MEGAN offers many plots and different visualizations. You can also always export your data and do further analyses in tools such as R or MATLAB or Python. So MEGAN is really flexible in that way. Now, MEGAN comes in two different varieties. So there's the free community edition, but there's also the ultimate edition. And the main differences are that the ultimate edition comes with KEGG Pathways built in and a command line mode. So KEGG Pathways went commercial a couple of years ago, but they're a very good tool to explore biochemical pathways, and then from a metagenomics point of view, comparing those pathways between samples. Also, the command line mode is really useful because even though the primary use of MEGAN is that you can interactively analyze your data, the command line mode allows you to basically script your workflows so that you can run the same analysis for many, many samples or even studies. So this basically combines the best of both worlds. So the classical bioinformatics pipeline where you script everything, and a classical GUI program where you just interactively click on things and run your analysis that way. So with MEGAN, MEGAN is offers the world's fastest metagenomics because it integrates really well with the Diamond protein aligner. So Diamond is a tool that is able to generate a speed up of up to 22,000 times versus BLAST-X, which is the tool you'd use to compare nucleotide sequences to a protein reference database. And I'm not going to go into into technical details here at all. The really important thing is that Diamond is still being actively developed, and that you can expect further speedups in the future. And also, for example, currently the team is working on making Diamond run better on compute clusters so that you cannot just use the all the CPUs on one cluster, but you can calculate your alignments across the whole cluster at once. Yes, and in combination with MEGAN, so you get down to species or even strain level. On a functional view, you get your genes, but in combination with KEGG, you can also analyze metabolic pathways. This is a snapshot of a window you'd see in your MEGAN metagenomics analyzer. So this is a taxonomic tree. This is currently the NCBI backbone tree, and overlaid on top of this tree is the relative read abundance across 4 samples in this case. And what you can do is you can do a first visual comparison here of the relative abundance of those bacteria across your 4 samples. You can collapse or uncollapse the tree, and the tool will then add up all the reads that sit up, that belong to a higher up node here, for example, to the family level, or you can uncollapse and look at the taxonomy or the reads across the taxonomy at a more granular, finer granular level. Of course, what many people want to do is they want to investigate the microbiome function, as as a lot of people have talked about already, because function is really important, and function seems to be well more conserved than the actual taxa that contribute those functions to the microbiome. So this is an example view here where you see one biochemical pathway, and then the read abundance across your different 4 samples in this example. And when you look at this enzyme down here, enzyme commission 4211, you can see that this enzyme is present or is supported by reads from this blue and this yellow sample, but the middle samples don't have that enzyme. And a slightly different scenario here for this enzyme down here. This is not present in the first sample, but it's present in all the other samples. So this is an example of a functional analysis based on a KEGG pathway, but MEGAN also works with Go annotation, eggnog, and eggnog. Okay, so MEGAN is used for visualization and exploration, but MEGAN doesn't do the alignments itself. So the alignments of your query sequences, so your sequence samples, they still have to be generated externally. And this is the preferred workflow that we have. So you obtain your sequencing data for your samples. When you're working with short read data, you obtain your FASTQ files immediately. But if you work with long read data, you have to do some file conversions, but the workflow is essentially the same. And the analysis pipeline works well for short Illumina reads and long PacBio or Oxford Nanopore reads. You obtain alignments by aligning your reads against the large NCBI non-redundant database using the diamond aligner. And those alignments then need to be meganized. Now, this is a tool you'd run on the same server that you've used to calculate those alignments as well. Meganizing essentially just means counting the reads that support each taxon in your data and tabulating that. Now, depending on the performance of your desktop computer that you would use for your MEGAN analysis, you would either load those alignments straight into MEGAN, or you can do another quick step, which is you calculate a comparison file, which is a very lightweight representation of all the genes and gene functions, taxa and gene functions in your data, and then load that into MEGAN. And then all you need to do is basically supply your study metadata, which is, for example, information on your treatment groups, if you have such a study, or collection sites, or whatever you need to make sense out of your samples. And then you can immediately start your interactive data analysis and explore your data. Okay, very brief update from 2 weeks ago. So a new version of MEGAN was released, and now KEGG has been updated to the very most recent version. So now is really good, if you're interested, or if you're already a MEGAN user, now is really a good time to do an update and start your analysis. All the other lookup tables and databases have also been updated. So the NCBI taxonomy, the seed functional database, the eggnog gene cluster database, so all been updated. And a new feature has been introduced, which is classification based on the GTDB taxonomy. That taxonomy is a genomics-based taxonomy, so that's computed on bacterial genomes, and so it's more a phylogeny than a taxonomy. So if that's your reference taxonomy database of choice, MEGAN is now able to work with this. Okay, so I've talked about MEGAN now, and now I'm going to switch to MORPHEUS. And MORPHEUS is also a product for the analysis of metagenomics data. So the question is, how is MORPHEUS different from MEGAN? And with MORPHEUS, we are aiming to simplify the analysis even further. So with that, I mean, we want to make it even more accessible for users and researchers. So MORPHEUS offers a set of standardized analyses that almost every user will need. And then you can always add customized analyses on top. We're using the same technologies as MEGAN also uses. So we're using MEGAN in this workflow, and we're also using the diamond aligner, which is why those tools will work seamlessly together. And one of the big benefits is you don't need local compute infrastructure to run MORPHEUS. So all the like the heavy calculations that you need for metagenomics, this system can take care of all that. You need a web browser to access our server, and that's about all you need. We'll also offer, we also have curated publicly available databases, metagenomics microbiome databases that you can compare your own samples to. So this is how this system works. On the right-hand side, we've got clients. On the left side, we've got a sequencing lab. Now, if you're a client that has your own sequencing lab, that's fine. We're assuming here for this graph that you as a client, you ship your samples to a sequencing lab. They will perform standardized DNA extraction and library prep and perform the DNA sequencing, and then you get your raw sequencing data. Now, your data is then transferred to our MORPHEUS servers. Depending on how we set the system up for you, this can also be done automatically. So it can go straight from your sequencing provider to our servers so that there's no copying from the user side. And then a standard set of analyses is triggered. So the servers will run a set of standard analyses, and they will also store your data for you. You can then choose to either download the data and plug it straight into MEGAN and do your analyses local, or you can log onto the server and interactively view the results on the server. Now, you need to provide some study metadata to make sense of your samples. So you need to describe the treatment groups, for example, and then you can immediately start viewing at our visualizations and going through the interactive reports. So the aim here is really that at the point where the researcher, the client, sees their samples for the first time, they don't have to, handle massive gigabytes or larger files and run analyses, but instead, when they see their data for the first time, it's already been analyzed and it's already results. So they get to view results, and they get to see information. And this is difficult to explain, so I'll try and do a live demo of this system. However, I need to, yeah, okay. So, okay, so this is basically, so when your samples have been processed from our servers, you get a notification, you get a demo, you get a login to our server, and this is here just a demo login to illustrate how this works. And then you are presented with this start screen, and you have a couple of apps available to interact with your data. Now, the first thing you would do is you would use this app here to provide study metadata to then obtain meaningful visualizations, which I've already done here. But then you can go straight into the analysis apps, and I'm going to show you very briefly 3 of those apps now. So the first app basically is to investigate the most abundant taxa. So this demo user only has one study, so we're going to click on this study, and this is a representation of the most abundant phyla across those 36 samples. So every stacked bar is a sample, and every colored bar in here is a taxon. You can choose different levels of the taxonomic hierarchy here in your data set. So if we go to the genus level, you see all the genera identified in your samples. And then when it gets interesting is when you look down to the metadata that you have provided, and you can sort by your metadata. Now this example data here, we have two different diet groups. One is a high protein diet and one is a moderate protein diet. And I've just sorted by those diet groups so that all the samples from the high protein diet are on the left and the other diet group is on the right. And you can immediately see that there's differences between those two diet groups. And for example, this one here, this genus Megasphaera is highly abundant in this diet group and is not present in here. Now, remember, this is now 2 minutes after you've provided your metadata. So you've already learned something about your samples. So you've just been presented with your sequences, your sequencing results. You can already start investigating your data. There's a couple of features here. You can, hide taxa. You can do all sorts of things. You can get further information on those taxa by clicking on them. But yeah, I just want to say again, so almost immediately after logging in, you've already learned something about your samples. So I think this is a really great feature of this platform. So just very briefly, I'm going to show two other apps on here. So one is a PCA app that basically shows you the structure of your communities. And again, we've got different methods of coloring by metadata groups. And we can show different principal components here. But I think this view is sufficient to illustrate that in this example data set, we have two very distinct groups of samples on the left and on the right. The very last app I'm going to show here is around microbial diversity. There's a couple of diversity indices that have been pre-computed that you can choose to visualize. Let's go with the Shannon diversity here. And you can then group your samples by your different metadata columns. So in this case, it probably makes most sense to go by the diet groups, and we can see differences in diversity between those diet groups. So let me get back to my presentation. So to summarize this, with Morpheus, you get immediate visualization of taxonomy and gene function in your browser. You can then choose to download your data and plug it straight into MEGAN6, so you can perform further analyses. And but already on the server, you can start investigating your data and without running any complicated pipelines or even having to handle those large files. The whole platform is based on standardized file formats and bioinformatic and statistical pipelines. And this is why it's easy to extend it to either third-party applications, or if you have if you like this this kind of way of working with your data, but you think there's an app missing, it's relatively straightforward to add additional apps to the system. And our goal really here is to make the analysis of large shotgun sequencing files a breeze. And depending on your sequencing provider, we can guarantee turnaround times for the bioinformatics analysis, and usually your sequencing provider can also guarantee turnaround times. So if you have a time-critical project and you really need to make sure that your bioinformatics pipeline is finished by a certain date, we can offer that with this platform. Yeah, so that's already it. So if you're interested, my contact details are below, and you should also be able to find them on the conference website. Please go ahead, contact me for a free trial license for MEGAN Ultimate Edition and see if that works for you and the problems it can solve for you. If you're interested in a demo of the Morpheus platform, let me know. I can either give you a more in-depth demo, or I can give you login information that you can have a look on your own. We may even be able to put your own data onto the platform so you can see if it can give you further insights for your own data. Now, one thing that I haven't talked about until now is that we also do customized metagenomics data analysis. So a lot of customers, for them, we work to understand their specific research needs and help them set up their project and develop very tailor-made analysis pipelines for them. So this is something I haven't talked about today at all, but if you're interested in this, this is something we're also doing for our customers. So yeah, feel free to reach out to me, and thanks for your attention.
Okay, thank you very much. There is a question here. Can MEGAN be used to analyze data from human samples, such as stool or oral or stool microbiome data?
Yes, not a problem. So we can even, so with MEGAN, so it's completely agnostic of the host species. There's some added features, for example, filtering host data. So when you're working with microbiome samples, usually you want to concentrate on the microbes, and depending on your samples, there can be a lot of, in this case, the human DNA would be contamination, right? So there's features built in to filter out the host DNA. And yeah, it works with human, it works with environmental samples. Yeah.
Okay, excellent. Any other closing remarks? We have a couple minutes left if you'd like to add anything.
This was a bit early, but it was difficult to time the live presentation, and I wasn't even sure, the live demo, and I wasn't even sure if it was going to work really well.
Oh, it worked perfectly, and you were right on the minute. So I thought we'd take advantage of it.
George, we have 2 more questions in the chat section, if you can check.
Okay, thank you. I didn't see those somewhere else. Here we go. Thank you. So how much does it cost for the MORPHEUS platform? What's the pricing like?
I think that's something we'd have to discuss because it really depends on the number of setup that's required. So if this is, we can install this at your site, and this is obviously going to be a bit more expensive, or you can have a per study or per sample model, which would be way more affordable. But this would require further discussions. So feel free to reach out to me. I'm happy to explain all the details and the costing. Yeah.
And thank you. And how long does the testing take?
Testing in terms of
Not sure if that. I'm not sure if that was going for here. That's in our queue here. Yeah, it wouldn't really be testing. So that must have been for a different one. Sorry, that came in just a minute ago. Well, I think that's it. So thank you very, very much for your time and your presentation. That was great. Very impressive work too.
Thanks, George.
Okay.